Welcome to our lecture series, part 9, ultraviolet spectroscopy, and we are explaining the instrumentation as a part 1, 2, 3, and 4. So in the previous parts, we discussed the instrumentation and we discussed the light source, what are the properties that the light source should have, and what light source we are using for this ultraviolet spectrum. We discussed hydrogen deuterated lamp is the most suitable one below 375 nanometers, and above 375 nanometers, halogen lamp is a suitable one. Then we discussed about the dispersive elements. The dispersive elements may be a grating or a prism, which splits the light into a several wavelength, and the monochromator, which helps us to filter the light of a required wavelength. And there is an entrance and exit slit. Now, in this session, we have to discuss about the sample container. We usually call this as a cuvette, and the detectors. The sample and the reference cells. So when the light comes out of the slit, exit slit, the light will be in two beams. One beam passes through the sample solution and another beam passes through a reference like this. So two beams, one will passes through the reference and another will passes through the sample. Both the sample and reference solution is contained in the cell. The cells are made up of a silica or quartz. This glass cannot be used for the ordinary glass cannot be used as a cell because the ordinary glass can absorb the ultraviolet light. So this is a special type of cell. We call it as a cuvette. It is made up of a quartz or silica, which will not absorb any ultraviolet. So you have to fill the sample in this, and one cuvette should contain a reference thing. So when the light source, after coming to a monochromator, it's split into two ones, and one as stated above, it passes to the reference, and the second one, it passes to the sample. So what happens here is, when it passes through the reference, no absorption can take place, and it passes to the sample, absorption takes place. So the detector, generally we have a photocell, and one of the photocells receives the beams from the sample, and the second detector receives the beam from the reference. The intensity of the radiation from the reference cell is stronger than the beam of the sample cell, because in the reference no absorption takes place, but in sample the absorption takes place. This results in the generation of pulsating or alternating current in the photocells. The photocell works in the concept of photoelectric effect. When a light of a desired intensity falls on a metal surface, it emits electrons. So detector, usually a photomultiplier tube, is extensively used to detect the ultraviolet instruments. When it falls over there, the electrical signals have been developed. Then the electrical signals are amplified by this amplifier and sent it to the recorder. The alternating current generated in the photocell is transferred to the amplifiers. So amplifier is coupled to a small servometer, and generally the current generated in the photocells is of very low intensity. The main purpose of the amplifier is to amplify the signals many times so that we can get a clear and recordable signals. So by recording these signals in the recording device, that gives the data to the that gives data through the computer. It stores or it gives in any other format like a Word file, Excel, or Notepad. So data can be processed to understand the type of a compound and where the lambda maximum absorption takes place so that we can understand the type of chromophores. So here, as a part of instrumentation, light source, monochromator, which involves entrance slit, exit slit, dispersive element, maybe a prism or a grating, and a sample container, reference container, detector, and amplifier. So the whole things is considered to be the instrumentation of ultraviolet spectroscopy. Thank you. In the next session, we will discuss about the application of ultraviolet spectroscopy.